Okay. So I'd like to talk about recent data that we collected on a project looking, taking a slightly different perspective, and which is addressing the question of what are the mechanisms which link exposure to germ cells. So, oops. Guess you don't see the title. Okay. I don't know how to get rid of this window. Is it on your way as well?
It looks fine on our end.
It's fine. Okay. So the question is really how, upon exposure, a common denominator in epigenetic inheritance, whether intergenerational or transgenerational in particular, is that the germ cells, the gametes, are affected. But what are the mechanisms which link exposure to these gametes? This may be very different depending on the type of exposure. For endocrine disruptors, like we just heard from Mike and Victor, bisphenol A or vinclozolin or DDT, the way they may be acting directly on germ cells via via estrogen receptors or androgen receptors can be quite well circumscribed. So it's possible that these components act directly on germ cells. But for complex exposure like stress and trauma, this is much more complicated because stress and trauma will induce many different things in the body, will alter stress hormone, will change behavior, will alter metabolism. So there is not one single component which may explain how germ cells end up being altered in the epigenome. So I'd like to suggest one possibility, which is the contribution of blood. So the idea is that blood composition is altered by exposure, and that there are components in blood which may induce changes in the epigenome, in epigenetic factors everywhere in the body, including in germ cells. So we don't know what these components could be, but it could be RNA, proteins, metabolites, or a combination of these factors. So it's the question that we have looked at. But it's quite complicated because exposure, there is not one kind of exposure. Exposure can happen at different times during development, prenatal life, childhood or adulthood. Exposure can be short, can be long. So if it's a long exposure, for instance, it will hit different developmental stage of germ cells. And as Victor showed you, so germ cells, they develop constantly, and it's very different if there is exposure, if there is stress exposure during prenatal life when there is only precursor germ cells compared to stress exposure in adult life when there are spermatogonia and sperm cells and spermatogenesis is going on. So we have started to address this question by focusing on childhood. Why childhood? Because in neurobiology or in psychiatry, childhood is a very critical period, and there are many psychiatric disorders which are induced or caused, or which are the consequence of traumatic experiences, being traumatic events, violence, abuse, physical abuse, sexual abuse, abandon, neglect in childhood, and which lead to symptoms which persist across life, and very often diseases like depression, antisocial behaviors, personality disorders, which are associated with childhood trauma, and the comorbidities, including cardiovascular diseases and cancer. They run in families, and despite many genome-wide association studies, there is not a single gene for depression, for instance, but it's a really combination of genetic background and the environment. So we are interested in how being exposed to trauma, childhood trauma, can affect the body and can alter the epigenome in germ cells. So I'd like to go through three questions. The main one is whether circulating factors can influence germ cells. So some of you may know of the Weissman barrier, which suggested it was really a hypothesis. It was not supported by any data at the time by August Weissman, which suggested or hypothesized that soma or somatic cells cannot communicate with germ cells. So in theory, this question may look a little incongruous. So what we did to address this question was to capitalize on a mouse model of postnatal trauma that we developed in the lab now almost 20 years ago. So it's a model which is inspired from the human literature and which combines maternal separation, so unpredictable maternal separation. So mouse pups are separated from their mother every day, 3 hours per day, from birth to 2 weeks postnatal. And this is combined with unpredictable maternal stress. So we place the mother during separation on either one of these stress, restrained stress or forced swing. So it's really a combination of physical, emotional, physiological trauma. And we look at the consequences across generations, so always in parallel with control. So we have MSUS mice, and we breed them with wild type females to generate the F1, F2, F3, and we have now generated up to the sixth generation through the Patry line. We also produced a Matry line, but until the second generation only. But this manipulation, this childhood trauma, this stress, we have found that it alters behavior and it induces the risk-taking. It increases risk-taking behaviors across generations. So here it's a compilation of many years of work showing that increased risk-taking that we evaluate on one of these tasks, which is the elevated plus maze. As you see here, this animal is taking a lot of risk. And normally a control mouse will stay in these closed arms. So, and this behavior is transmitted through the fathers, through the Patry line, up to the sixth generation. The animals are also depressive, as expected from the human literature. I just show quickly this video. So stress coping or depressive-like behaviors can be tested on post-salt force-swing tasks. So you see here a control mouse, a depressed mouse which floats. And we see this depressive phenotype up to the third generation. It's not present anymore in the fourth, fifth, or the sixth generation. In addition to these behavioral alterations, there are many metabolic dysfunctions. Also, weight is altered at the beginning until the F1, F2 are decreased weight in adulthood, and then there is an increased weight. Somehow as a rebound. We cannot really explain why this is opposite, but I see you've seen from Mike and Victor that the phenotype can evolve across generations. And glucose metabolism is also altered in F2. Here, upon restrained strain, you see restrained stress, you see that glucose mounts, but it's always lower in the MSUS descendants of animals directly exposed to the trauma. Not in F3, but then it comes back in F4. So globally, behavioral alterations, metabolic alterations, and we have done a lot more assessment of these of the behaviors that I don't have time to go through tonight. So the question can circulating factors influence germ cells? To address this question, we collected serum from MSUS mice. So you see here the MSUS and controls. We collected serum in adults and collected blood in adults, prepared serum, and injected serum to control mice IV twice a week for 1 month. So the idea was that if serum contains factors which may communicate with germ cells, after injecting these adult males with serum, breeding them to generate an offspring, it could be that we may be able to see symptoms of trauma in these offspring, which is the offspring of males injected with a serum. And that's exactly what we see. You see that we could, so these data here on the left in green are MSUS, and on the right in yellow are the animals resulting from the injection of MSUS serum into males. So you see that the reduced weight is could be recapitulated, as well as the decreased glucose response in response to restraint. So indeed, serum injected chronically into a control mouse can send signals to germ cells, which are then inducing some phenotypes in the offspring. Now, the question is which blood components are altered by exposure and could explain this communication with germ cells. So to address this question, we conducted unbiased proteomic and metabolomic analysis on the blood of the animals. And after really many months of work and data mining, we came up with the observation that the major metabolites which were altered are linked to lipid metabolism. And in particular, alpha-linolenic polyunsaturated fatty acid metabolism. So the metabolites which are derived from these polyunsaturated fatty acids, as well as arachidonic acid metabolism, bile acid biosynthesis, and steroidogenesis. So while this is decreased here in pups, which were after 8 days of trauma, when these pups are grown adult, so many months after the trauma, we see that there is an increase in the metabolism of polysaturated fatty acids, and to some extent, some of these alterations are maintained in the offspring. So now, is this relevant at all for trauma? So to answer this question, we collected a cohort of children who have been exposed to trauma. And for this, it's Ali Javaid, who used to be a PhD student in my lab and then has left the lab to have his own a couple of months ago. So he's originally from Pakistan. So what he did was to contact the SOS Children Village Administration, and he went to Lahore, his hometown, many months, several years in a row for a couple of months, and assembled a cohort of children who have lost their father and were separated from their mother, to be as close as possible to our MSUS model. And he collected blood and saliva and brought that back to Zürich, and we did similar mass spectrometry analysis, and we could see that in serum or saliva, we could observe a comparable alterations of lipid metabolism in these children. So somewhat validating some of these changes that we saw in animals. Now, the last question is which blood components, which of these alterations could signal to the germ cells? And in the polysaturated fatty acid metabolites, fatty acids are very interesting because they can bind to nuclear receptors and all these different components which we found altered in our metabolomic analysis. They can bind to different nuclear receptors, including PPAR and its cognate receptors, LXR, so liver X receptor, or farnesoid X receptor, which is a bile acid receptor, and which also associate with the retinoic X receptor. And the beauty about these receptors is that they provide an interface between something in blood and chromatin. So we postulated that perhaps these altered metabolites in blood could reach germ cells and modify gene expression, gene transcription, or chromatin structure in germ cells, possibly at different time during development. And one confirmation that PPAR may be involved is that its expression is increased in sperm of MSUS males. So suggesting that PPAR pathways indeed are altered by the manipulation, by the trauma. So then to really test this hypothesis in vivo, we used a PPAR agonist that we injected in control males, exactly the same way we injected serum. So twice a week for 4 weeks. And then after this, we waited 46 days for a full spermatogenic cycle to go on. And then we crossed these injected males with naive females, generated an offspring. and then waited until they were adults, test the behavior and the metabolites, metabolism, and then breed them again to generate a grand-offspring. So we wanted to see whether activating PPAR with this agonist in adulthood could have an effect, could lead to a signal, could signal to the germ cells, and whether they could eventually pass over 2 generations. And indeed, we saw that in both the direct offspring and the grand-offspring, adult weight was decreased and glucose was reduced on a glucose tolerance test in both generations. So suggesting that PPAR activation and PPAR pathways may be one of the pathways that contribute, that link these alterations in lipid metabolism to germ cells in vivo. A last set of data is this really relevant for germ cells? Because here it's in vivo, and it could be an indirect effect. So what we started to do in vitro is to use spermatogenic-like cells, GC1 cells, and the idea was to treat them with serum or to treat them with TESA and see whether we can reproduce the effect. So what we first did to address the question of whether serum in germ cells acts via PPAR pathways. So we took these cells, transfecting them with a PPAR reporter, PPAR again being a nuclear receptor which acts as a transcription factor. So once these cells were transfected, we treat them with serum, and then we measure luminescence, so which is a direct measure of PPAR activation. And we could observe that if we use serum from MSUS males, this activates PPAR, the reporter, PPAR reporter. And if we use serum from TESA injected males, as expected, because TESA is a PPAR agonist, we also see an increase in PPAR activity. If we now, you may ask, well, what is the link with the epigenome? Is it possible? What does PPAR activation or serum itself, possibly through PPAR, to germ cells, to the epigenome in germ cells? So we have started to address this question by measuring the level of microRNAs in GC1 cells after treatment by serum from either MSUS mice or from the traumatized children. And we could see that so GC1 cells treated with serum from MSUS mice, there is an increase in microRNA 375, which we knew before to be associated with a trauma and being altered in this MSUS mice. And likewise, the serum from traumatized kids could increase MER 375, but also alter another microRNA 16. So all this put together suggests that serum can act on germ cells, and that may be through PPAR. So in conclusions, serum from trauma-exposed males into control males can recapitulate some of the metabolic symptoms in the offspring, suggesting signaling through the germline, and that this signaling involves lipid metabolism and PPAR pathways, probably not only PPAR pathways, but PPAR pathways is involved in this signaling. And all of this suggests that there are possible common routes of communication with the germline via metabolites, because PPAR has been, as Victor said, PPAR has been associated with transgenerational transmission linked to obesity. For instance, there was a very nice paper by the lab of Bruce Bloomberg a couple of years ago on the effect of an obesogen compound. So I'd like just to advertise our paper is just published today. So for those of you who want more detail, I refer you to the manuscript. So I'd like to finish by thanking people involved. So this is the work of Gretchen van Steenwyk, who did all the metabolomic analysis and identified PPAR signaling. Cathy Gap, who was a former PhD student in my lab many years ago, who is now back to Zurich after spending some time in Cambridge, took care of the serum injection and all the serum analysis. Ali, who took care of all the human analysis and collection of the samples from the skid and the assembly of this cohort. And the bioinfo was conducted by Deepak Tanva and Pierre-Luc Germain. And I would like to thank also our collaborators, Eric Miska, Nicolas Zamboni for all the metabolic analysis and proteomic analysis, which I did not show, and the SOS Children's Village and Mrs. Sabah Faisal for allowing us to work with these children. And I thank you very much for your attention.